Hello everyone. So today I Swati Verma, Assistant Professor at Calcutta's University School of Medical and Allied Sciences, shall be discussing with you about the introduction to UV-visible spectroscopy. So in our previous lectures we have discussed about the basic of spectroscopic techniques, that is what is a spectroscopy, the different types of spectroscopic techniques and the various functions of spectroscopy techniques. Now we'll discuss each and every spectroscopic technique in detail. So first we will discuss about our UV visible spectroscopy. So moving forward, first we will discuss about what is UV visible spectroscopy and what it does. So the strength of UV here refers to your ultraviolet. So the strength of our ultraviolet radiation lies in the ability to measure the extent of multiple bonds or aromatic conjugation. So what our UV radiation do? Our UV radiation will measure the extent, that is the number of multiple bonds or your aromatic conjugation which are present in a particular molecules. Now what is conjugation? Conjugation is the presence of your double bonds at the alternate position. That is, we can have your double bond, single bond, double bond. So we have presence of double bond at the alternate position, and this particular conjugation is measured by your UV. It is also known as electronic spectroscopy, as your UV visible spectroscopy involves the transition at the electronic level. In the previous lecture, I have told you that our total energy comprises of your E electronic, E vibrational and E rotational. So your UV radiation, being the highly energetic, causes changes at the electronic energy. So it involves electrons and produces changes at the electronic level. So this is known as electronic spectroscopy. It involves the promotion of electrons from the ground state to the excited state. Now it measures the number of conjugated double bond and also aromatic conjugation within your various molecules. So with the help of UV visible spectroscopy, we can measure the number of conjugated double bond present. It also distinguishes between conjugated and your non-conjugated system. We can have two systems, conjugated and non-conjugated. In one we have the presence of conjugation and the other we don't have any conjugation present. So on the basis of that we can differentiate between these two. Next, we can differentiate between your alpha beta unsaturated carbonyl compounds from your beta gamma analog, as well as we can differentiate between your two dienes, that is homoannular and heteroannular dienes. And what are these? These we will discuss later on when we will have a discussion about Woodward-Fisher rule. So for UV and visible spectrum, the electronic excitation occurs in the range of 200 to 800. So this is the range of our UV visible excitation, and it involves promotion of electron to the higher energy molecular orbital. So upon UV visible absorption, a molecule which is present at the lower energy level will show a transition to the higher energy molecular orbital. Now we'll discuss about the excitation process, that is how the excitation of electrons from the ground state to the excited state occurs. So in case of UV and visible spectroscopy, the transition that results in the absorption of electromagnetic radiation in this particular region of the spectrum are transitions between your electronic energy levels. So your UV visible radiation, or you can say UV visible spectroscopy, involves transition at the electronic energy level. So as your molecules absorbs energy, electrons are promoted from your occupied orbital to an unoccupied orbital which is present at your greater potential energy. Your unoccupied orbital, that is your excited state, is having higher potential energy. Your UV region is further subdivided into near UV, which is below 200 nanometer, and it starts from 185 to 400 nanometer, to your far UV, which is from 400 to 700 nanometer. Next, we will have a discussion about your wavelength ranges of UV-visible spectroscopy. So here is your broad detailed discussion or description about your wavelength ranges of UV-visible spectroscopy. So in the first we have your regions, and then the second we have your wavelength ranges mentioned in nanometer. So the first region that we have is your vacuum ultraviolet region. Your vacuum ultraviolet region starts from 10 nanometer and ends to your 200 nanometer. This particular vacuum ultraviolet region involves transitions of your single bond, and this particular region is not accessible in each and every UV visible spectrophotometer. Then we have near ultraviolet region. Near ultraviolet region will have a wavelength range of 200 to 380. Next we have visible region. So visible region is further divided into these. First is your violet 380 to 435, blue 435 to 480, green blue 480 to 500, green 500 to 560, yellow green from 560 to 580, yellow from 580 to 595, orange from 595 to 650, and then red from 650 to 780. Here I have written certain colors in brackets. So within bracket, the color which is mentioned as the color of the radiation which is absorbed by your molecules. So the absorption of yellow-green radiation occurs at 380 to 435, and the color of the radiation appears is your violet. This is your transmitted color and this is your absorbed color. Now this is your detailed excitation process, which we will understand by means of this particular diagram. So here I have mentioned the ground state and this is your excited state. So the molecules are present at your ground state. So whenever we will give our energy of electromagnetic radiation to our molecule, what will happen? Your molecules will show a transition from the ground state to the excited state. So upon absorption of UV visible radiation, your molecules will show a transition from the ground state to the excited state. Now there is a particular criteria by which this particular absorption occur, or you can say a requirement. The requirement is that the energy of your incoming UV visible radiation must match to the energy of this particular delta E. Now what is delta E? Delta E is the energy difference between your two states, that is your excited state and the ground state. So your incoming electromagnetic radiation must match to the delta E. Your delta E is the minimum amount of energy required by your molecule so that they can show transition from the ground state to the excited state. Now next we will discuss in detail about what is absorption of electromagnetic radiation. So as you all know that the basic principle of UV visible spectroscopy is absorption, that is your molecule will absorb the incoming UV visible radiation and will show transition from the ground state to the excited state. Now in case of absorbance spectroscopy, a beam of electromagnetic radiation passes through a sample. So we will impose a beam of electromagnetic radiation to a sample. When that particular beam of radiation passes through a sample, much of the radiation is transmitted without a loss in intensity. So as if we are having 100% intense radiation, so the much of the radiation passes through a sample is having the same particular intensity. But at certain selected frequency, what will happen? We have the radiation intensity attenuated. That is, we get a decrease in the intensity of incoming radiation, and that particular decrease in the intensity of incoming radiation is known as absorption. So basically we can say what is absorption? Absorption is the decrease in the intensity of incoming radiation. So a molecule will absorb the incoming electromagnetic radiation, and for that what will happen? The incoming radiation intensity get decreased, and after absorption your molecules will show a transition from the ground state to the excited state. So there are general two requirements which are to be met if an analyte is to absorb your incoming electromagnetic radiation. The first requirement is that there must be a mechanism by which the radiation's electric and magnetic field interact with the analyte. So we know that the electromagnetic radiation is having the electric and magnetic field. So that particular electric and magnetic field must interact with the analyte. The second requirement is that the energy of electromagnetic radiation must be similar in energy between the two energy levels. So the energy of incoming electromagnetic radiation must be equal to or similar to your delta E. Delta E is the difference between your energy states. Moving forward, we have the nature of electromagnetic electronic excitation. So whenever a continuous radiation passes through a transparent material, what will happen? A portion of the radiation must be absorbed. If that particular absorption occurs, the residual radiation which is then passed through a prism yields a spectrum which is having certain gaps in it, and that particular spectrum is known as absorption spectrum. So our absorption spectrum is having certain gaps in it. As a result of energy absorption, what will happen? Your atoms and molecules will pass from a state of lower energy to a state of higher energy. So after absorption of incoming UV visible radiation, our atoms or molecules will pass from a state of lower energy to a state of higher energy. In case of UVVisible spectroscopy, the transition that results in the absorption of incoming radiation in this particular region of the spectrum are the transitions between electronic energy levels. So the transition that we have in this particular case are the transitions of electronic energy level. So we can say that the UVVisible spectroscopy involves electronic level transitions. So whenever a molecule will absorb your energy, an electron which is present at your ground state will be promoted from an occupied orbital to an unoccupied orbital which is of greater potential energy. So this particular transition is known as a transition from HOMO to LUMO. So here HOMO refers to highest occupied molecular orbital and LUMO refers to lowest unoccupied molecular orbital. As our excited state is your unoccupied orbital, so we can say that this particular transition is from HOMO to LUMO, highest occupied molecular orbital, to your lowest unoccupied molecular orbital. For most of the molecules, the lowest energy occupied molecular orbital is the sigma orbital. We have different types of orbital. So the lowest energy occupied orbital is the sigma orbital, which corresponds to sigma bond. The pi orbital, the next type of orbital, is lie at somewhat higher energy level, and these particular orbitals will hold your unshared pairs of electrons. The non-bonding orbitals are even present at the higher energy level. So we have three types of electrons present. We have sigma orbitals, pi orbitals, and the n orbitals. On the basis of that, we have three different types of transition occurring: your sigma-sigma transition, your n to sigma star transition, your pi to pi star transition, and your n to pi star transition. These are your certain references that you can refer. These are certain names of the books for detailed description about your various types of transition, and to study what is UVVisible spectroscopy, you can refer these. Class, thank you so much.